Thanks, Martha, for the kind introduction. Yeah, the title seems a bit enigmatic, but actually, since Corona, I've begun to think of phage genomes more and more as a monopoly board, and how are we trying to understand it? So basically, when we compare the monopoly board and a phage genome, of course, we don't know most about it. I think 60% of the genes, so 60% of all the streets are not known, a lot of regulatory elements. But what we do know is that there is a very strict organization and regulation. It's clear, hopefully, at first glance, when you sequence a phage genome, what is the organization, and you logically assume how things are regulated from early to middle and late gene expression. Of course, this can be impacted by some weird genes that get inserted, and you have some horizontal gene transfer. But how does that impact that overall regulation? Well, it seems to accommodate this pretty well as part of the board game. As you all know, phages have very densely coded genomes. There are a few intergenic spaces, like free parking on the monopoly board. But is there really? I'm going to go in that as well. And I really consider every gene in there, because we're working in a confined space of the phage capsids, that this genome is genetic real estate, and every gene that's in there, even though it's essential or not, it's through billions of years of coevolution has been carefully selected for, and it's there for a reason. So I consider it all to be important real estate. And what I want to talk to you today about primarily is a sequencing technique called ONT-capable-seq, which is an easy RNA-seq approach that is specifically aimed to be low-tech and to elucidate the transcriptional landscape of microbial viruses and find out some new things about it, in addition to another tool that I will be talking about. So Oxford nanopore sequencing has been mentioned a few times. It's a nice technique. The principal point behind this is that it enables long read sequencing by pushing the nucleic acid strand through a pore, and then differences in conductivity are being measured and interpreted. And over the past few years, there have been a lot of improvements in terms of library prep and chemistry. A lot of work has been done on, sorry, that's missing, in terms of base calling, in interpreting these differences in conductivity and improving the sequencing run overall. And it's estimated that the newer kit is going to have more accuracy due to a slower translocation throughout the alpha hemolysin pore of the nanopore, and increasing accuracy further. But actually, the main trait that we need for our ONT-capable-seq is the long read. And with all this in mind, our people are very happy to play around with this Promethean. It's in parallel. So you know the mid-ions. This is the Promethean, which has a couple. It's nice to see how you can smile even with a mouth mask. Yeah. So we're very enthusiastic about it, and I'll explain why. So what do we want to do with this ONT-capable-seq? When you consider the phage transcriptome in its basic element, you can have a promoter, some genes, a terminator, and a single transcript, a primary transcript, will be processed further down into the final molecules that are being made. But those primary transcripts are of specific interest to us here because they define the transcription start, the transcription stop, and the sequence in between. And this can be quite lengthy, so that's the advantage of tracking this with nanopore sequencing. So those primary transcripts constitute only a small percentage of the RNA molecules that are inside. and we need a way to get them out. And interestingly, the triphosphate end of those primary transcripts are unique to them. So using a specific capping enzyme, you can cap biotinylation here that next you can, with the streptavidin beads, can be specifically pulled out and then sequenced through long read sequencing technology. So how does the workflow look like? We have our capping reaction, our poly A tail gets added, then we can have a split of the sample between the two. One sample we're going to enrich for the streptavidin, and then we do our first strand synthesis, a second enrichment step, our second strand synthesis, the PCR barcoding and amplification to have the longer reads ready after adapting ligation to put on the machine. Can I get rid of the little thing? No, sorry? This one? Perfect, thanks. It's a magic trick, huh? That's probably the most information you take from this talk. So RNA, usually you have the big two ribosomal RNAs after you have your control and your enriched sample. After sequencing the control versus the enriched, we could actually show that there is a huge increase in the amount of phage and also bacterial coding sequences within those samples once you capture them. This was applied to an N4-like phage called LUS7. It's actually quite interesting one because this phage co-injects a giant RNA polymerase that expresses the early genes. Then there is a multi-subunit RNA polymerase encoded here that is going to be responsible for DNA metabolism. And the final genes are actually expressed by the host RNA polymerase with a phage-encoded sigma factor. So we're not sure about many of these promoter elements that were there. Even though there is, sorry, the colors are quite light, but you can still within the genome, you can capture what is host takeover, what is replication, what is virion assembly and lysis. From there, we wanted to see, can we actually get the promoter identification that we need. With classical RNA-seq, on your genome, you're going to compile your reads. They're nice and blue on my screen, but still you can make it out. In any case, capturing those reads, you see the sequencing depth here as well. And actually with the ONT-capable-seq, you get much more striated reads that are coming in, allowing you to actually see your transcription start sites and stop sites. So we went further with this, discovering specific motifs that weren't observed before, that were experimentally confirmed, and using traditional primer extension, we could actually confirm some of these. And this primer extension, to do that is very time intensive, and that's the traditional way to do that one by one. But here we're capturing all of them at the same time. And to further confirm that these promoters were active, we had a simple vector system where the phage promoter was placed upstream of the GFP. And then we could actually measure the amount of GFP that's being produced from that specific phage promoter, driven by specific RNA polymerase in question. So those transcription starts are really nice. It allows you to experimentally validate those promoters present in your board, in your organization of your transcription. one master student said, hey, you're covering all of the coding sequences from the phage, but can't we pull these transcription start sites also from the bacteria? And indeed, that's possible. And during his master dissertation, he took the data from ONT-capable-seq, also some differential RNA-seq that we did together with the Vogel lab, to extract the transcription start sites, and immediately upstream should be the promoter motifs that could be selected, and then actually assuming that these are correct, yeah, and this is an experimental validated promoters, can we use those in modeling, setting up a neural network to be trained using those promoters as input, and then implement this in software to be used. And that's what we did. That's what's illustrated here, simply for some sigma 70 promoters that were pulled out in front of the transcription start sites. And they were predicted some -30 and some -10 sites, and combined and selected. Each dot is actually one promoter element that was extracted from the bacteria. And these were actually used as canonical examples of the sigma 70 that would enable different models to be trained and see what the sensitivity and specificity and loss was for predictors. So what does that mean? Once we have these the best models and the best predictors, we can go actually into a software where now you can input your sequence either for Pseudomonas and for Salmonella, and you can put in your genome and just say, okay, where are the sigma 70s, and identify those. That's just one example that you can do for Pseudomonas, for Salmonella, and that specificity is needed to have more accurate models that are doing well. In our case, the best models were these convolutional neural networks that were applied. And when we tested them on samples and compared it to other established promoter prediction tools, the latest version is outperforming those more generalized systems. And I think that's the basic take-home message. Rather than having one size fits all promoter prediction tool, we have to go towards species-specific predictors. And if that's driven by RNA-seq data trained on assumed or predicted or verified promoter elements, that will make it easier and more reliable when you annotate your genomes. That was all about the transcription start sites, and the promoters were equally interested actually on how transcription terminates. And so the transcription termination sites, how do they get stopped? And one interesting case here in that LUS7 genome, where we had a very strong promoter, and we identified a number of terminators that were very weak. Yeah. You see here the pullback in the number of reads and sequencing depth. So this was sort of a stepwise transcription termination pattern. So we're hoping, I don't think this is an experimental artifact. What it could mean is that these not so strong terminators may be involved in the stoichiometry of how your proteins and the specific ratios of your proteins, for example, to compile your virion particle, are going to be governed. You can think about this as a way that it's transcription-based modulation of the amount of proteins that are there. Obviously, this is also going to be impacted by codon usage and so much more. But all of it, nothing will be left to chance. And everything will be subtly optimized to get those optimal ratios for the proteins that are needed. Another example we observed is when we were taking these capable-seq samples early or middle and late. And we saw that even actually the way we predict some of these transcription start and stop sites are actually in the drop-off rate in the number of reads that is observed. And here we actually saw that early in transcription, these reads were going further, and during middle transcription, this was somehow more potent in terms of the number of reads you see there. So again, there a subtle regulation of terminators that is going to govern the ratio and presence of proteins. And even here on a 4 base pair space, there were reads in both directions, and they went to a full stop. And these were very potent bidirectional transcription terminator terminators that we're also looking into to see if we can utilize those in genetic circuits for symbio applications. We wanted to experimentally validate also these phage-encoded terminators. So made this nice little circuit of a promoter that's driving GFP. In between is a terminator followed by M-cherry, where you can look at the ratio between GFP and M-cherry to ascertain the strength of your terminator. The stronger your terminator, the less M-cherry will be present. And we also observed that the terminator is actually influencing the amount of protein that GFP that was generated upstream there again. So that gives us an entire array of terminators with a very flexible activity range or termination potential, let's say. By the way, those little circuits, they're being pieced together in a very easy way. We set up this system called Seva-tile. That means that each individual part is cloned with tags on the side to enable very easy combination in a single pot reaction. So it's sort of like golden gates and versatile shuffling for protein engineering. But here it's used to piece together individual parts to create these genetic circuits. And Evelyn Marie is working on that. And that allows you very easily to have hundreds of or thousands of these terminators to be tested in parallel and to create circuits as you see fit. Another aspect beyond transcription start and stop sites is that we're also seeing small RNAs. This is a gap here in the genome between ORF69 and 70. There were no genes predicted, so this was predicted as free parking space within the genome. That's not the case. We clearly saw transcripts being present, promoter driving them, terminators being present. So these small RNAs that are often overlooked and largely not seen on your phage genome monopoly board, they are there and they will serve a purpose because they've been selected for, they've allocated genomic space for them, so they have to be serving specific roles. We also seen some antisense RNA that will also likely be involved. So going beyond genomics and having these transcription information is going to very much fine-tune your genomic landscape at this transcriptional level. And indeed, I'm not a believer of anything just happening for a reason on that phage genome. It's always going to have a purpose. One small final example is a T7-like phage for Pseudomonas we isolated recently, called LUS100. It's Autographiviridae, and this is always considered to be the prototype for lytic phages. If we think about T7, very easy, transcribed from one side, left to right, with host takeover, genome replication genes, virion assembly genes, and lysis genes neatly organized in the board, except for this one, this 100 here. And Jason Gill saw something similar, that there is in fact something happening here in front of the RNA polymerase. We identified an integrase. So meaning that there's T7 phages out there that are not strictly lytic. And yeah, that can be a scary thought because these phages are often used. So diligence, even in the phages we think we know well, should be always considered, thinking about the previous talk. So if we zoom in on this, and of course we used our ONT-capable-seq to identify promoter elements, terminator elements. We found all of them, I think, knowing the architecture of T7-like phages. We got the strength out of it, but we wanted to see what's happening specifically in this lysogeny part of the genome. I should mention we haven't been able to isolate stable lysogens from them, but what we think is happening here based on the ONT-capable-seq is the following. Yeah? We have the RNA polymerase, which is the principal driver of T7-like phages transcription early during infection. Ahead of this is an integrase and also a MAR-R type transcriptional regulator. So what we saw, and this is schematic of course, is that transcription levels of this integrase and transcriptional regulator are high, and there is some read-through of the phage RNA polymerase. So going into the lytic cycle, at some point this will be high enough to really trigger phage RNA polymerase transcription, and this is going to be a self-amplifying loop with the first phage promoter there, and that's going to further trigger expression of early genes, DNA metabolism genes, and structural and lysis genes. Yeah, and what we're figuring out now is that probably this MAR-R-like transcriptional regulator is going to function as a switch to sort of block and keep this phage as a lysogen under certain specific conditions, I believe, from blocking RNA polymerase from being expressed. So we're also getting some functional transcription regulation clues from those transcriptomics data that we want to look into. So overall, for us, this ONT-capable-seq is a very straightforward approach to get all of these elements while serving also as a validation of your gene predictions, right? The transcripts you have covering genes will validate those as well. It's inexpensive. I think when we run a few samples on the Promethean, it's about 150 euros. So that's not too much. And we've applied it now to some Pseudomonas syringae phages, Thermophilus, Cellulophage baltica, and some Salmonella species with Lone Brunsted's group. That doesn't mean that this technique at all replaces your traditional RNA-seq. This is rough and dirty, so to speak. There's no statistics involved. I mean, replicates involved. We don't do quantitative analysis. We don't go for an actual host response and seeing how phage infection expression levels in the bacteria. We're not doing anything quantitative along those lines. If that's what you're looking for, you of course need to go back to the traditional RNA-seq. But it does allow you to go well beyond simply genome sequencing based interpretation of how your transcription regulation is actually functioning, And we think that having these maps can also be a tool for antibacterial and biotech applications inspired by phage, not in the least if you have engineered a phage and added all kinds of equipment. How well is that machine running? How well is that transcription map going? These are things you can think about and see, is my design actually functioning? And if you're going to design phages or re-engineer them, having these promoter elements available or having additional information on how the operons and so forth function is likely going to increase the success rate at which you're generating your phages. I want to thank my group, Lena, Jorien, and Maarten on the ONT-capable-seq and the LUS 100. Lucas, who is doing a PhD at Imperial and did continue this as a hobby for the Sapphire. I'm good at convincing people to do that. And Evelyn Marie on the Seva tile, and my entire crew